Hello everyone, my name is Kevin McKernan and I'm here to present on Cannabis Genomics in 2015. We had the great honor of having 3 abstracts selected for presentation at the International Cannabinoid Research Society, or the ICRS 2015, and we wanted to re-record these because we did receive permission to record them, but the audio quality we captured on iPhones didn't quite capture the sentiment or the detail of a lot of the scientific slides. And so I'm going to be presenting on sequencing of 3 male cannabis genomes and the development of multiplex qPCR assays. And these assays are for rapid male sex determination. And why is sex determination important? Well, half the plants come out as male, a little bit over half, and the other half come out as female. And females produce drug and flower and medicine and seeds, and males tend to be used for other types of qualities in the plant, like fiber and growing tall, thin hemp plants. So there's a reason to be able to distinguish these at a very early stage. Currently, in large cannabis grows, you have to wait 6 to 8 weeks to figure out what is male and female and cull them. This culling process can be error-prone, and if one male sticks around, it can pollinate an entire grow and destroy hundreds of thousands, if not millions of dollars of crop. For this reason, many people prefer to clone the plant. And cloning the plant certainly can, at least if it's a mother plant, can get around this problem, but it does bring on the challenge of monoculture. When you begin to clone plants, you tend to have genetically identical plants, which are then perhaps more prone to disease. And so some people would love to have the benefit of diversity in their grows, that are all seeds from the same parents, but perhaps slightly different, enough to give them some resistance, as opposed to clones of a single mother plant. And so the topic of doing sex determination is in fact controversial. Some people would really love to use seeds if they had a good way of guaranteeing they're all, in fact, female seeds. And there is a whole field of seed feminization and a science behind that. There are pros and cons to this. Some are concerned that the feminization process can in fact create mutants. And it is in fact it is an art as to how to do that. So we're going to talk about male sex determination in this presentation, and how we can potentially change this with the advent of next generation sequencing. Now, I have some conflicts to declare in that I am an employee of Medicinal Genomics and Corrigan Life Sciences. I'm a CSO of both companies. They're really one company, all under the umbrella of Corrigan Life Sciences. The Medicinal Genomics side is just the plant genetics. We focus on cannabis plant and the genetics of the cannabis plant. And the Corrigan Life Sciences group focuses on the healthcare implications and the genetics of the endocannabinoid system. And so they are a CLIA and CAP certified company that sequences the genomes of patients that have epilepsy, autism, mitochondrial disease, lysosomal storage disease, and any drug-drug interaction really with the pharmacogenomics that they do. So that's very much the human side of the endocannabinoid system, or the ECS system, and medicinal genomics is very much the plant-based side of it. So with those conflicts declared, I certainly have my own personal conflicts, in that I'm a male individual talking about Y chromosomes in the plant, and as many would know, males have a stereotype of not being able to read other people's maps, and that has certainly happened in this case. There is great work done by Giuseppe Mandolino on finding male PCR markers that can determine male from female in cannabis. The challenge with these markers is that they create a complex banding pattern that you can see in this picture here. This banding pattern has to look for this little tiny band that distinguishes male from female, and if you want to automate this with something like quantitative PCR that gives you a digital yes-no answer in a 96 or 3D4-well format, well, you can't be having people stare at these bands. This is going to be a challenging thing to automatically screen. And so we set out, somewhat arrogantly, to convert this assay into a qPCR assay like you might see here on the left, and thought we'd have the job done just replicating the work in a matter of a month, and this, of course, turned into a much more complicated onion that just had more and more layers every time we dug deep. Now, we shouldn't have been too surprised by this because they did give us a pretty adequate warning. This MAD-C2 region that they targeted was shown quite elegantly in their own publication to be a repeat. And we probably didn't quite appreciate that this repeat might exist in more than 2 copies that are shown here, that it could in fact be replicated manyfold more than what was known at the time. So we confidently ran about replicating their primers, and we sequenced all of these bands that are produced here to try to figure out if there was a sequence unique to the smallest band that we could qPCR. Now, right there should have been a red herring, an alarm, trying to find the sequence of something that's clearly repetitive and shares primer sites amongst all of these bands in the shortest product is hard to do, because the shortest product by definition has the least unique material and it shares the same primer sites. That's proven out to be the case and very difficult. What we were finding was that these primers weren't very reproducible in the genetics that we had. And I want to underscore that latter point, is they were very reproducible in prior published work, but I think the prior published work may have been focused on a subset of genetics that perhaps aren't in circulation today. So we did, in the midst of getting tangled in this big onion, decided to sequence the entire genome of a clone known as Wi-Fi, which is a male clone. And this showed that we had about 17,000x coverage when we only aimed to get 30x coverage in the genome. So you sequence the genome to 30x coverage, and the one region that has 17,000x is actually MAD-C2. So that's another warning sign that, okay, there could be about 500 copies of this MAD-C2 region in the genome, even in male genomes. And so what is it going to look like in females? And when we looked at this in females, there was an equivalent number of copies. And so what you're looking at here is a set of reads, sequencing reads, often an Illumina platform. And the red reads are reads going in one direction on the DNA strand. The green ones are going in another direction on the DNA strand. And all of the vertical highlighted letters are areas where they see single nucleotide differences, or what we call SNPs, single nucleotide polymorphisms. And you can see over on the right, the reverse primer actually has SNPs underneath the primer, and that's fairly dangerous to have when you're designing any type of qPCR assay, for there to be polymorphisms under your primer sites, because it means there is going to be a plant out there in the population that has something you probably don't anticipate and won't prime and will leave you with a false negative or potentially a false positive. And if you look at the other side of this region, what you can see is there's actually, on the left-hand side, there's actually a SNP on the 3' end of the primer. That is the most important base to get right. The polymerase very much scrutinizes that base, and if that base is different, it's going to differentially amplify one thing or another. And we do, in fact, see a few reads in here that are blue when you look at them horizontally, and those are perfectly paired reads, meaning the forward and reverse reads are the expected distance apart, and they in fact represent the right expected 400 base pair, 390 base pair product that we're looking for. Nevertheless, there's still SNPs in some of those, and so this is another concern. You will notice a lot of coverage from the reads over on this side, on the left side of this region that we've mapped, and then a kind of a grayed out region in between, and that's because most of these forward and reverse reads do not agree on the insert size that we're mapping to. So this is known as what's called a broken paired read. We have forward and reverse reads that don't agree with the fact that we told it that we should map this to about 390 bases, and they're highlighting the fact that they appear to be more distant. That's expected to happen in a repeat that differs in size, just like Mandolino demonstrated. So what does this mean looking at a PCR assay? Well, before we threw in the towel on using this set, this primer site as an assay, you have to recall that when you want to build a quantitative PCR assay, you're not just relying on the 2 external primers. In fact, I'll go back once just to show this. You're also relying on trying to place a probe somewhere in the middle that will differentiate the male from the female regions that are amplifying. And this looks like a very messy region to design an internal probe for, because there's so much polymorphism right between the 2 primers. So we asked ourselves, is there any way we can potentially improve this using different temperatures in the annealing time and the annealing temperature of PCR. For those not familiar with PCR, at a particular time when you're annealing your primers to the target DNA, that temperature and time is very important for specificity. And as you go higher in temperature, you get more specificity because as you increase heat into that equation, it becomes more and more difficult for those primers to anneal unless the match is perfect. And so people often do gradient PCR to bring out more specific priming sites and to figure out the ideal and optimal temperature at which something will prime. Typically, when you increase the temperature in a gradient PCR, you get more and more stringent PCR, i.e. less bands show up, and you tend to hone in on the right temperature that amplifies your desired band. Well, we're not seeing that with the MAD-C2 primers. What we see is we increase temperature, which as you can see going from left to right in this graph, that we tend to get more and more bands as we increase the temperature. So this means we can't optimize our way out of the problem with simply changing temperature. We really have to screen this against a diverse set of drug-type strains and preferably even hemp-type strains so that we can find a temperature that's going to fit the entire genetic population. And that to us seemed to be a bit challenging. So what we decided to do, since we'd already sequenced an entire male genome known as Wi-Fi, why don't we back ourselves out of this problem by throwing more sequencing horsepower at this and just sequence truckloads of plants to identify better Y chromosome markers, ones that aren't repetitive, preferably. And so in this experiment, what we did is we sequenced 12 different females. Females are XX and males are XY. And we sequenced 2 different males, a third to verify, but 2 that we're going to discuss initially. And then what we did is we took all of the reads that we had from the male genome project and we filtered them against the females. And anything that didn't stick to the females, we deemed putative Y. We would save those putative Y sequences, and we do some filtration. The first filtration you'd want to do is make sure that these aren't just microbes that grow on the plant. There's a whole host of microbes that can grow in cannabis, and there's a whole other presentation on that in the Medicinal Genomics website about the microbiomes that grow in cannabis. But obviously those are candidate things that won't match the female references, so you got to filter out those. We definitely want to filter out repeats. Repeats are of no value to us. They're very difficult for us to utilize as markers because repeats are some of the most active regions of the genome, and they tend to move around on you. This is Barbara McClintoff's work, and it's quite known that transposition is kind of the GM of modern day normal plants. They actually have quite plastic genomes when you look at the transposition frequencies. And then we also want to make sure there's no polymorphisms because the Y should be homozygous. And to the extent that you end up finding diploid regions, you want to avoid them because they're probably not on the Y. They're probably crossed over with the X, and they share 2 copies, and that's probably going to be a dirty marker. And then you of course, screen for good GC content and other amplification criteria with tools like Primer3. So we designed a handful of amplicons to test out these putative Y contigs after assembling them to come up with a hypothesis. Now, for those not familiar with this process, it's pretty simple. You just need 30 to 50 nanograms of DNA from the plant. You can get that from a Sensitivix prep that we currently, Medicinal Genomics actually sells on their website. It's a very easy magnetic bead-based method to purify DNA that basically just takes some trichomes off the plant or off the leaf or off the stems, and you can get DNA off of any of those tissues with this kit. And you typically get about 30 nanograms to a microgram of DNA, but all we need is 30 nanograms to run a NextGen reaction, which is a transposition system that fragments that genomic DNA into smaller pieces that are amenable for sequencing. Now, the most important part of DNA sequence assembly is that you have some understanding of the distances between your reads. We can get, thankfully, 250 base pair reads off of Illumina sequencers these days, and we can get those by the hundreds of millions, if not to billions. If those reads are just randomly used to try and put together the cannabis genome, you'll have a bit of a mess. What you really need are paired 250 base pair reads, where you know the distance that they sit apart. And ideally, there's a very long distance between those two reads. In the case of Illumina, you can get them to be about 800 bases to 1,000 bases apart by size selecting them and clustering only those. A little bit difficult to get them any further than that on the current platform, but paired 250 base pair reads, when they're known to be about 800 to 1,000 bases apart, very much helps in positioning the data in a repetitive genome. So just to point out that Akagi's actually done this on Lotus, although they didn't have a reference sequence like we have in Cannabis. As a result of not having that reference, they had to use many more strains to get to this answer. But we have some references, and we can get away doing a few less genomes to sort of this problem. So we ended up doing about 80 million reads to, well, it looks like 88 million, 98 million, and 80 million for Australian Bastard, Grape Stomper, and Wi-Fi. These paired 250 base pair reads we then use to do that filtration process and come up with a putative 2,326 unique contigs, combining all of the information for the three strains, and some preliminary blast analysis can be performed to rule out things like Homo sapien contamination you might see in here. There's also, what you want to look for is that there aren't any hits to the nearest organisms like hops, because the nearest organism sequence like hops, they have only sequenced female hops lines to date. So if we found any hits to hops, we know those were putative female. We're glad not to see that. We see what is grape and citrus and a few other lotus species that we can then decide whether these things are male or female based on what they might look like in other genomes. So after all that filtration, we can then boil this down to a binary answer, a quantitative PCR assay that gives us one band, not many. And the reason that's important, as I mentioned earlier, is if your PCR product produces an array of different bands, you're always going to need an electrophoresis system tied in front of it to figure out if it's male or female, and we don't want that. We just want to measure the PCR real time and get the answer many steps ahead of having to amplify it, purify it, put it on a CE instrument, and figure out what the proper band size is. We're trying to reduce this to something that's potentially field portable and can work at the step of PCR, which is the first step that you would perform the moment you had genomic DNA. So what we're looking for is a yes-no answer, something that's more blue-green, or if you want to say blue-red, like you might get in a typical diagnostic test. We need a single band or no band, and that's in fact what we're getting with the primers that we've designed. So what we then have to set out to do is we have to validate this. The cannabis plant is so polymorphic that it's important that you validate this on geographically distinct strains and on as many of them as you can get your hands on, which turns out not to be very trivial considering males are highly selected against in the cannabis field. Okay, so here's a case of grabbing 10 samples out of Maine, and we can see that we have two different signals coming up in qPCR, and that's an important point. We have a signal for the Y chromosome, and then we have an autosomal signal known as SCCG. That stands for single copy cannabis gene. It's a gene that's in the cannabinoid synthase pathway. That turned out to be a harder assay to optimize than the Y because the single copy cannabis gene being in the cannabinoid synthase pathway, that part of the pathway is tenfold more polymorphic than any other region of the genome. And it makes for a difficult time designing primers to be able to afford all of those different variations. So we've had to screen that amplicon across several hundred different DNAs to make sure that it works. And there's some very intricate insertions and deletions in that region that we had to dance around. The Y has only been screened to date across about, in this presentation you'll see about 41 of those samples. We have many more coming in the door as we speak, but it's just harder and harder to find male plants these days. So there's a whole other presentation on this SCCG, but the reason this is important is that we want to make sure that the DNA prep always worked, and the PCR has to always produce at least one signal for you to believe it. And if that signal happens to be extraordinarily low, in other words, it tends to come up past 35 cycles, in this case you're seeing most of them come up at around 30 to 31. If it comes up that late, we're not confident that we actually, at the Y, had enough time come up and signal, and we may have just had too little DNA out of the prep and missed the Y. So this is a really important measure to let you know that you got enough DNA out of your prep and you know that the PCR is working. And only if you see SSCG can you then make a comment on whether Y is there or not. And so then we moved on to do this in Seattle and grabbed another 10 strains there, and all of those performed successfully. And then we went into the Humboldt County region and grabbed another 20. And when we started summing all of these things together, we ended up getting about 40 out of 41 males successfully amplifying. So one male showed no Y signal. Now, we didn't have any pollen sacs to confirm this was a male. A lot of this is material that's DNA given to us secondhand, so we only get a picture of maybe a leaf, and you can't tell sex from a leaf. So maybe this is a sample handling error on our part. We don't know. What we were doing is taking that sample, graduating it to next generation sequencing, and figuring out, is it just an artifact local to this one amplicon, or do we see the other 2,300 amplicons lighting up with signal as well? If we see that, then we're pretty confident it's a male that we missed. And that data is currently crunching on sequencers. We did have 2 flowered hermaphrodites that tested as female, which, knowing that they're flowers, they should. That's a very interesting attribute to the assay. And then we had another 40 out of 41 females test as female, another 65 out of 65 females test as females. So there's 1 female that tested as a male, and we're whole genome sequencing that as well. Maybe we just sample swap this one with the other, and that's the issue. Now, we have to keep in mind that calling a female falsely, let's say if you get a false positive on a female and you call it a male, that's never going to kill anybody. It's not a very large economic burden if you happen to falsely call a female a male. If you fail to pick up a male, however, that's a male that can sneak through and do a lot of damage to a very large grow. And so we're far more worried about missing males than we are falsely calling females. And whenever that's the case, you can design your specificity and sensitivity assay in your favor. So we're whole genome sequencing these discordant males. They're slowly starting to trickle through, but the real answer as to whether or not these probes and primers are going to behave is to basically go deep into the most distant landraces that you can find and see if the primers are conserved into distant landraces. And the most distant landrace that we could find is a topic of a whole other presentation at ICRS that we were fortunately granted a presentation for, known as Australian Bastard Cannabis. This is a strain that came out of Australia. It's believed to be extraordinarily different. There's a whole other presentation describing this. It has phyllotaxy. It has a very different leaf structure. You would not notice this to be cannabis, although it does interbreed with cannabis. And we do know that it in fact has a Y chromosome marker, the one that we're targeting, and it does amplify for SCCG. Now, just some caveats on this. This strain that we are handling has been crossed with the sativa, so it is an Australian Bastard strain that's been crossed with a sativa. So there are some sativa genetics in there that are probably responsible for this Y and this SSCG signal we're getting. And to really sort this out, we need to sequence the entire plant of the Australian Bastard clone, and that's precisely what we've gone on to do, and that is the topic of the second presentation for ICRS. Now, there's a handful of folks that deserve credit for this. There is many people that have helped us in the field, and just to teach us about cannabis and sexing and all the history that's been done here, including the folks down in the helpful communication bucket down here. There is a very large Cortigen team that has been generously giving their time to ensure that these assays run efficiently and effectively and according to some of the best standards in the industry. The Cortigen team is a clinical sequencing team that makes tests that diagnose patients, and so they take sequencing accuracy into the most serious category you can imagine. And then, of course, we have over 120 investors that have invested in this privately, and we're very thankful for their trust in this area, including a reggae band known as Slightly Stupid, which we're very grateful for their in-kind music donations to some of the apps and the websites that we've been building. And we're always encouraged by any type of popular support for the work that we do. But most importantly, really is a team of very dedicated individuals up here at the top of the list that have been endlessly fighting to make better products in this field. And so with that, we will turn it over to the second part of the presentation, which is known as is there this in fact a fourth species of cannabis with the Australian Bastard clone? Thank you.